episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Right. Welcome, everybody, again to another fascinating episode with another guest that is involved in creating a better tomorrow for all of us. We're honored to be joined today by Dr. Ilan Murad, who is the CEO and founder of a startup company called Accelerated Evolution Biotechnologies. Dr. Murad has his master's and his PhD from Tel-Aviv University in biochemistry. He has a background in a variety of aspects of drug development and drug discovery processes, including gene cloning, protein engineering, purification and characterization, various aspects of the drug discovery process, and most recently has been actively involved in developing a really fascinating, what I'll call a combinatorial biologic technology for developing therapeutics for multiple targets simultaneously, called Multiple Targeted Safe Comprehensive Cancer Therapy Platform, or Mutato, and we're going to dive into a lot of that. Dr. Murad is located right now over in Israel, so we will now join him. Dr. Murad, thank you for taking the time to come on the show today and talk to us for a little while.
Okay, thank you for having me. It all started actually 20 years ago when we started the company. We had an idea about a screening platform, and after a few years of working on this project, we were able to have it done. Now we have patents for this screening platform, and we use this screening platform to discover novel therapeutic peptides for the diversity of illnesses. And now we use it in this project of cure for cancer. So about 5 years ago, we started this project. And since we worked on cancer before, we had the feeling that what we're doing is not enough, because we had such a great tool in our hands, and we should do something better than just me too, find another drug that would give you more years or months to cancer patients. So we start thinking about something much bigger, and we wanted to find out why most of the cancer treatments do not actually cure the patient. Some of them do, but in most cases, they don't. So there's a big problem there, and we try to understand what is there are many problems, but we wanted to understand the full picture.
Yep.
And it took us actually a few years of experiments to understand it. And actually, it's not so complicated. The main problem with cancer is that the cancer cells are very mutagenic. And because of this mutation that they can acquire very easily, they can adopt to many situations, including therapy. So they can escape the therapy by acquiring mutations very easily. And that's the main reason why they escape therapy and get the drug resistance. Of course, there are many other reasons, but this is the main reason. Now,
before you go deeper into that, I just want you to take a couple minutes, if you would, because we've gone into a little bit into this topic previously, but I want you to stress it. I think it's extremely important, and that is when you talk about cancer cells being mutagenic, we don't always think of a tumor as, you know, a lot of times we think of it, or the past has taught us it's this runaway cell growth. But when you really look at a tumor nowadays with the 2020 hat on, it's not one cell. It is a really heterogenetic mix of stuff going on, and it's why you can kill off certain cells that may have one mutation, but there's other cells in that population that have other abilities. Talk a little bit about, when you talk about how cells are mutagenic, talk about sort of the degree of this heterogeneity that we understand tumors to be nowadays, and why then multi-target toxin targeting and so forth is so relevant. Take us on a little bit of that path.
Yeah. So, you know, every cancer cell starts as a normal cell. Then it starts accumulating mutations. And by the time it becomes a full-grown cancer cell, it may have dozens or hundreds of mutations. Now, if you look at all of the possible mutations, there are thousands, maybe even more possibilities. So, if you talk about a solid tumor, it starts from one cell and then starts growing. And when it grows and grows, it accumulates more and more mutations. So, if you look from the inside towards the peripheral, you see that you get more and more mutations towards the periphery of the tumor. Now, if you look at the whole tumor, you see a lot of variants of the original cells. Now, each mutation can cause something. So probably some of the cells are already drug resistant to several drugs. Maybe most of them would not be, but if you use the drug, you kill all the, like the same thing as with antibiotics. You kill the cells that are not sufficiently stable towards antibiotics, and you're left with the ones that are resistant to the antibiotics. Then they are growing freely, and then all of the cells are resistant to antibiotics, and then the story is
right.
complete. Same thing with cancer. So when you try to kill the cancer cells, you should kill all of the cells and all of the variants, and not just some of them. And this is probably the main reason why many treatments do not succeed or succeed in lower percentage. So as we think this is the main problem, and we thought that we have to find a solution towards this problem, and we think that we do. I mean, when we were hit by this fact, it immediately let us remember me personally, a remembrance a long time ago, like 20, 25 years ago, when I first heard of the AIDS cocktail. Yep. I said, oh, what a genius idea. I mean, it's so simple, but a genius. Instead of attacking only one HIV target, why don't we attack 3 at a time? And then the whole thing was solved. Now the cocktail saves people's life. Now people actually, if you take the cocktail, nobody should die from this disease.
Right. So
it's the same thing. The HIV is very mutagenic. This is why when they used only one drug, it didn't help for more than a few months. And then the HIV mutated, and again, it developed drug resistance. Mm-hmm. So when they started using the cocktail with only 3 different drugs, it solved the problem. And it's all statistics, because every creature, it may be a virus, HIV, or it may be a cancer cell. It follows the statistics. So creating one mutation is pretty easy if the cancer cell or HIV are very mutagenic. Mm-hmm. Now, 2, it's much harder for them, because statistically, to get 2 mutations, specific, not just any mutation, specific mutation that would fight the treatment, is much harder. And then if you talk about 3, it's almost impossible for them. So when they use a cocktail of 3 in the AIDS, they solve the problem, and the same thing should be here. And this is exactly what we do. So our Mutato contains at least 3 targeting peptides. Each one of them attacks a different cancer target. This is one thing, but we added a twist. We added a 4th peptide, which is a toxin. Now, this is very important. Not only that the toxin, okay, let's talk about the toxin. The toxin is inert outside of the cells, because its target is inside the cell. So when each one of the targeting peptides interact with its cancer target, it makes internalization or endocytosis. Then the whole Mutato gets into the cell. Now the toxic peptide can find its target and kill very efficiently the cancer cells. Now it's important not because of another way of killing by a different mechanism, but there's a very important thing. You know, most of the mutations in cancer cells are internal mutations. So if you talk about, for instance, a signal transduction, where you pass the signal that comes from the outside through the receptor and towards the nucleus, there are a lot of several messengers there. Now, mutation in any one of them could stop the whole process and make the treatment not useful. So if you can introduce another peptide, like a toxic peptide, into the cell, and then the peptide kills the cancer cell by another mechanism, then it doesn't matter if you have 100 of internal mutations, because all this Mutato needs is to get into the cells. When it's inside the cell, it can kill by another mechanism. So many mutations can be overcome by introducing this toxin. So altogether, it is almost impossible for the cancer cell to overcome such a treatment. I mean, attacking at the same time 3 cancer targets, which are receptors, which lie on the outer membrane of the cancer cell, plus the toxin. Now, people criticized us by saying, okay, so there are thousands of mutations, so you can't fight all of them. But we don't have to fight all of them, because, you know, when the protocol of using the treatment is first having the biopsy of the patients, checking it out to see which receptors are overexpressed, and maybe even using the same peptide that would attack the cancer cells as for the analysis. So when we do that, we know from the beginning which receptors respond, and then we know that there isn't a mutation that disable this interaction. So we start from a situation that we know that works. Now the cancer cell should adopt mutations to overcome this. So the cancer cell should acquire at least 3 mutations to overcome this treatment, and this is almost impossible. So this is the basics of this idea. Now, there's another very important thing, because we use all the peptides connected through a multi-arm scaffold. So it's one molecule. Now, when you attack different targets and you do it from one molecule, there is what is called an avidity effect. Now, this avidity effect, what it makes is something like, if you attack one target, you get a specific one second. You get a specific I forgot the word, effectivity or efficacy.
Sure.
Now, if you connect 2 and not just give it as a mix, the avidity effect makes it much more effective exponentially. And now, if you add the 3rd one, it would be even much better than that. So we can say that in order to kill the cancer cells, if you use a 3 targeting peptide on a Mutato, and you compare it to only 1, then you would be able to kill the cells by a much lower concentration. And if you use lower concentration, then it would give much less side effects. So by using this concept, not only that the Mutato won't let the cancer cells escape the therapy, but this therapy would have much lower side effects at the same time. So I think it's a win-win situation. Now, we made many experiments using many kind of cancer cells and targets, using all kinds of different mutatos. We show, first of all, that we can kill many kinds of cancer cells by using the peptides that we develop using our platform.
Mm-hmm.
Second, we show this avidity effect using our Mutato. So as I just explained, and we did a lot of in vitro experiments. Then we moved on to show on whole animals. So we did a few mice experiments, and we show that our Mutatos can reduce the size of the tumors in mice in a xenograft model. So actually, I think that it's pretty much proven that this concept can work. Now what we have to do is go to humans and show it there. So what we want to do is complete the preclinical trials.
Mm-hmm.
I mean, what we have to do is repeat these mice experiments, but GLP-wise, do it on larger animals,
Mm-hmm.
do all kinds of pharmacokinetics experiments. We did some, but we have to do it in a way that the regulator would approve it.
Sure.
So we already talked to a very famous company, I don't want to say the name, very, very well established, one of the best companies in the world. They gave us a program for doing all of this in less than a year. It cost a lot of money, which we don't have. So we try to raise these funds. So another thing that we have to do is to synthesize the first Mutato. We already know which one would be the first Mutato. It would be a treatment for lung, colon, and head and neck cancers. Now, not to all of the patients, but only the ones that would have the 3 receptors that on our first Mutato. But
Mm-hmm.
I guess that would be a lot of them because we chose these targets. The principle that we followed was targets that are their existence are in many cancer patients.
Mm-hmm.
So we expect to that many patients would have these 3 targets. So the first Mutato would apply to many patients.
Mm-hmm.
So our first goal is to bring this first Mutato to the clinical trial and start treating patients. And our second goal is to develop 100 peptides, anti-cancer peptides, that combinations of these peptides, and that would be 1 million. Okay, so these 1 million combinations would cover almost all cancers.
And
so,
yet, to me, it's a fascinating. I mean, it fits in exactly with what we know cancer therapy should look like. It's personalized. It's multi-targeted. Talk, if you would, because we did you mentioned Mutato, but there's also this SOAP platform that it's based on, which is based on phage display. We did an episode actually last week on bacteriophages as therapeutics. Talk about how phage display fits into sort of what you're doing and how that technology could help in the personalization. Yeah. I mean, in other words, I want you to take this in the direction, if I'm thinking about this the right way, where, as you mentioned, there's so many different types of mutations we know about, we've cataloged over the years. How personalized could this actually get, in the sense that, boom, I do a biopsy and I show up one day at the Mutato Center, and you can make a personalized, between the SOAP platform and the multi-targeting, a personalized therapeutic from me versus the next person. Take us a little bit on that part of it, if you would.
Okay, first of all, you don't have to use the SOAP platform to discover novel peptides or whatever to use in the Mutato concept or in other concepts.
Okay.
The thing is that we own this platform.
Okay.
We have patents all over the world for this platform, and we can use it. I have freedom of operation. I mean, nobody can tell me, oh, you use my technology, don't do that, or pay me a lot of money that I can. So this is the most important thing, that this platform is ours, so we can use it as much as we want for any program. Now, it works, the platform works, and we did other programs not connected to cancer, and many cancer projects. I mean, to find anti-cancer cancer peptides. Now, again, the SOAP platform doesn't have to be used for cancer. We can use it for any project that we want. Actually, we thought to use it in the COVID-19, but we didn't have the budget to do that. But we can do it. If we have the budget, we can do it. So we use this platform to discover novel peptides against cancer targets and to use it in the Mutato concept. Mm-hmm. This is the connection. Okay? This is it doesn't have to be, again, we can use other technology to discover novel therapeutic peptides for the mutatos, or we can use the SOAP technology to discover other peptides to other projects, not cancer. Mm-hmm. Mm-hmm. But the connection is that we use it for this project because it is the best thing for us. We can do it easily, it's ours, we have all the things that we need, so we can do it very quickly.
What's next? You mentioned the preclinical development program. You mentioned the first indication. Other indications you're thinking about with regard to oncology that you're excited about?
Oh, we are working on other targets. So mm-hmm. we work now on HER2, which is for breast cancer.
Sure.
And we work on VGFR, which is also for breast, but also others. And there's a whole list that we can of targets that we can use and discover peptides towards them. And now, you know, we don't when we do that, we don't discover only one peptide per receptor. Mm-hmm. Our technology enables us to discover many peptides towards any target. So we have a whole panel of peptides to any target. Mm-hmm. Mm-hmm. So it's a lot of work, and we wanted to go on with this project, but we're a very small company, so we can't do it all by ourselves. It goes very slowly. Sure. So what we had in mind is, first, the most important thing is to get the first one as quickly as possible towards the clinical trial, because we want to start curing cancer patients, and we want to start with the most hard cases, like patients that no drug or treatment can help them anymore, and we want to treat them first, because we believe that if we can cure these patients, only a few, then the whole world would look at a different angle on us, because, you know, now many people are angry at us, and they remember our first interview in Jerusalem Post 2 years ago, and they said that we are cruel and we're not responsible, and what do you want? Many people said that without even looking at what we're saying or understanding, or even calling us, say, hey, why don't you clarify something? Right. So this is one of the reasons that last February, we worked on this article for 2 months, whole 2 months. We just sat and did nothing, just by writing this article. We wrote an article and published it in the Journal of Cancer Therapy. And this article, now I can, by a click of the hand, I can send it to anyone. It's common, everyone can see it. You don't have to pay anything to see the whole paper. And everything is there. Everyone can read the concept, see the theory, see our results, our proofs, and before you say that it's not good, okay? So now we have that. Everyone that said, oh, I don't understand, this is all kind of stupid stories, just send him the link, read the article first, and then talk to me, okay? So this is very important. I realize now that this is very important, because before that, there were all kinds of things that were told about our concept. Some people said that we use phage to cure patients, which is completely mixed up of all the facts. Some people said that it's it's a magic bullet that one drug would cure all patients, and this is not possible, of course. So many kinds of things. So the article is very important. Everyone wants to know more about and see about the Mutato concept and see results, how we kill cancer cells, how we reduce the tumor size in mice. Can read the article. Everything is there. So what we want to do next, again, first, most important thing is to complete the preclinical trials, start treating cancer patients in the clinic. This is the most important thing with the first Mutato. And while we're doing that, this is all by outsourcing. So we don't have to do anything, just pay other companies to do that.
Right.
Now, while we do that, we can develop more peptides to more cancer targets. So we want to have a list of 100. Again, 100 peptides to cancer targets. If we have this complete 100 peptides, I think that we can cover all cancers. And that would take a few years, but we have to start somewhere.
Sure.
So this is the complete plan.
Yeah, it's a fascinating plan. It definitely has all the pieces. I spent most of my 30+ years in the pharmaceutical industry, so I know why you got the feedback that you did. The drug world hates things that are not pure chemical entities that do one thing, and this is exactly the opposite of that. But it's what, it's, if we're going to cure cancer, it's what's required, and so you're on the right path. It's what's the innovation ecosystem like in Israel nowadays with everything going on with COVID, and how's the biotech system doing today?
Israel has a great biotech industry.
Yeah.
Many startups. They do all kinds of things. For instance, one of the companies, they have a treatment. You can say a treatment to to patients that in very bad situation, like half dead. Okay, they gave it to a few patients, and after 1.5 weeks they were cured. So for instance, but it's very expensive, this treatment. It uses cells, stem cells, and you have to grow them, and stem cells is very expensive.
Right.
So but it's a cure. I mean, all 100% of the people that they gave this treatment were cured. So this is one company, and there are all kinds of there's a vaccine, Israeli vaccine, that they work on. So they're starting. Of course, they're way behind others, but I think they're in their phase 2 now. And there are a lot of interesting biotech or health companies in Israel. And this is very good because we're supporting each other.
Yeah. It's a very impressive. In a past career, I was involved with some Israeli investors, and I was very impressed by the degree of the science that goes on there. So, it's a great platform, and like anything in biotech nowadays, money is obviously always the limiting step, but you definitely
it's our biggest problem.
Yeah, you'll definitely get there though with the way things are moving in this industry towards away from traditional drugs towards things that might not look like traditional therapeutics. This is spot on. So very exciting. Suma joined us. Suma, I see you over there. Sorry I didn't bring you in earlier, but how are you today? It's this evening.
Yeah, it's evening here. It's very nice to meet you, Ira, finally.
Nice to meet you too.
I pronounce it correct, right? It's Ira.
That's Ira. Yeah, perfect.
Hi.
I didn't get a chance, just as for the audience, we've been speaking to Dr. Murad. Ms. Suma Khazmo is a partner in this endeavor. She is the chief executive officer and co-founder of an organization called Regal Optics and Medical Devices, which is located in Ramallah in Palestine. Focuses on both importing and exporting metal devices into the Middle East and North African markets. She also is involved in the Regal-Medic Ophthalmology Clinic and the Hajal Optics Group, which has a chain of optometry centers in Palestine. She's also the chairman of the board of the Bayader Media and Publishing Corporation. She has a master's degree in international relations and bachelor's degree in media journalism and political science from Birzeit University, located in the West Bank near Ramallah. So how did you two get together? How are you involved in this project?
Involved with ABI.
Yeah.
Well, actually, I've been fascinated with the Mutato since a long time ago. So actually, I will tell you like a little bit of a story. I've been in the healthcare industry after, being involved in politics and international relations. And then I switched. I've always been in love with, biology, biotechnology since I was a kid. And then afterwards, I postponed it a little bit. And afterwards, when my father got sick with cancer, I was searching for treatments for him, actually. And it's not personal, but really I'm not being biased here. I've been looking at Mutato. I didn't know Dan or Ilan or anybody there. I just was reading the research. I always read the research. I don't look at, propaganda or just, articles in magazines. I just read the research. And actually also I read the research about you, Ira. You had very good research regarding other subjects I know of.
Sure.
And then it took me to Mutato. Mutato, it absorbed into my mind. It is a very great cancer cure that I'm fascinated with. And it's supported by science, very solid science, as Dr. Ilan has, clearly said here today. And it is peer-reviewed, it's patented. And I really wish that we could, push it to clinical trials, phase 1. So that we can help people, really. And, to be involved with such wonderful people like Ilan and Dan Aridor and Dr. Dan Pear, who's the chairman of the advisory board. It's very, very unique. We can learn from him every day, from all of the team at the ABI. It's very wonderful experience, hands-on, really. So
it's nice to see that, and obviously this is not a political show, but it's nice to see some I sit here obviously on the East Coast of the United States, and I have my limited view on the world, but it's nice to see that these types of things are going on in the region. Suma, you're very involved in startup businesses and angel investing and so forth in the region. Talk, if you would, a little bit just to educate us here, sort of in the U.S. Talk about the innovation ecosystem in Palestine, the Israeli-Palestine innovation relationship, because although we see what we see on the news, I'm sure there's a lot of interesting relationships like this, working on important things like cancer going on. Talk a little bit about that, if you would.
Yeah, actually what you see in the news, it's something, and what goes in biotechnology and science is something else. In every nation, there are the good people and the bad people, and there is war and there is peace. And, you can't say, for example, I hate stigmas, these people are bad,
Right.
Like Palestinians are bad or Israelis are bad. No, we are all humans. We have great minds, great brains. We have great scientists on both sides.
Yep.
So it is great that we can come together, in a peaceful relationship, in a scientific relationship to find cures. Even for cancer, for COVID-19, for anything, even in brain health, for the neurosciences, for everything that we always thought of as impossible to treat, we can treat it. And one of my mottos is nothing is impossible. I can spot great cures or great science, and I want to, what do they say, to adopt it. I want people to know about it. I want investors to come and invest their money in it. Actually, people say that in COVID-19, there is no money in the market, that, no, there is money, but investors are afraid to invest. So we must, you know, show them that we have something solid here. We have something scientifically based. We have something that can be also very profitable in the future on all sides. This is why. And my experience in the investment ecosystem has taught me that you must think out of the box, and that there are some investors who are also very open-minded, who want to invest in new things. Right, Ira? Correct?
Absolutely.
Because if we don't invest in new things, we cannot find our next Google. We cannot find our next Facebook. You know, we cannot find treatment for, let's say, brain death. We cannot find treatment for cancer. You know, people say like she's hallucinating or something like that. But no, we can. We can treat all of the diseases if we put ourselves to it. Correct?
Outstanding. Yeah, I'm 100% in agreement with you, and it's always interesting. As I said, I've spent most of my time in the pharmaceutical industry, but I had very nice investors from the region in the agricultural industry, which is obviously a very hot area in Israel and Palestine, and it's always interesting to see where there's that cross fertilization of opportunity. So, but just as a side note, because I was fascinated reading through your bio, you are in addition to being an entrepreneur in the area of ophthalmology and your involvement in startup companies and investing, you are also a very big advocate for animal rights in the region. Talk about that, if you would. Just tell us, because I didn't get a chance to fully introduce you at the beginning, but talk about how you got interested in animal welfare and what goes on at the Bethlehem Animal and Environment Association.
Yeah. Yeah, I work with a very wonderful person she's the founder of the Animal Association in Bethlehem, Animal and Environmental Association in Bethlehem. Actually, we try our best to save animals. We volunteer. We go to save them personally. We go on, we have this, what's it called, rescuing team, where everyone can just call us. We have, this injured animal, dog or cat or something like that, and we just go there and, and rescue it. Or we get, of course, donations, but they are not good enough. So we spend our own money on it. We have a shelter for animals. It's the first shelter in Palestine, by the way. And we try to raise funds to make a bigger one with, garden for animals where they can all the injured animals can come and, be treated. And then children can come and, play with the animals, for, what do you call it? To feel, connected with the environment and with the injured animals. Yeah, to teach children to love animals.
Yep.
Because if you love animals, you will love other human beings. This is the, the thing that I want all of the children in the world to know. And I've been working also on legislations for, the animals, the first animal welfare law in Palestine, of course. So it's good. I hope that we can succeed in this also. It is very important.
Yep.
This thing.
Yeah. If we teach
important. Animals are part of our ecosystem, are part of our world. We need to respect them. Yeah.
They teach empathy, and if we can take care of animals and love them, we can love one another, and that's a very important thing as well.
Yeah, I have 2 dogs, and they teach us very, very great, lessons about empathy, about tolerance. It's great. It's great to, to be a friend of an animal. It is. They are a part of the family. We need to respect them in order for this world to be better, really.
Absolutely.
This is my opinion.
Absolutely. Well, it's been really fascinating talking to both of you, and obviously I'm so glad that you two are working together on this to get these initiatives, both independently and together, moving in the region. For everybody that is going to be watching on the YouTube channel or listening on the podcast. You've been listening first to Dr. Ilan Murad, Chief Executive Officer and founder of Accelerated Evolution Biotechnologies, doing really amazing things with cancer and multi-targeted cancer therapies. Fascinating work. And Ms. Suma Khazmo, CEO and co-founder of Regal Optics and Medical Devices in Ramallah, Palestine. To both of you, thank you for taking the time. I know it's very late there. Thank you for taking the time out of your schedule to come talk to us about what you're both doing. Thank you for doing it. And as we say, thank you for helping to create a better tomorrow for the world and everything that's going to be coming out of these projects. It was really very fascinating. And appreciate you taking the time to come on the show and talk to us for a bit.
Thank you very much, Ira. We try to do our best, really.
No, it was great. It was really great.
Dr. Ilan, you wanted to say something? Yeah.
Please,
Sorry.
please.
Oh, I just want to thank you too. Okay, thank you.